Welcome, and thank you for joining us. My name is Dean Lee, and I'm Professor of Physics at the Facility for Rare Isotope Beams, or FRIB. Today's public talk is part of an initiative at FRIB called the Advanced Studies Gateway. The goal of the Advanced Studies Gateway is to inspire people. We bring together researchers, innovators, creative thinkers, artists, and performers from all fields, and strengthen ties between Michigan State University and the community. Today, we have the pleasure of presenting a public lecture by Professor Stephen Quake from Stanford University entitled, Understanding the Mysteries of the Cell. How do many cell types arise from one genome? Professor Quake is the Lee Otterson Professor of Bioengineering and Professor of Applied Physics at Stanford University. He joined Stanford in 2005, where he helped found and lead the university's bioengineering department as it expanded to nearly 24 faculty members. From 2006 to 2016, he also served as an investigator of the Howard Hughes Medical Institute. Professor Quake's research sits at the intersection of biology, physics, and technology development. He has invented a range of measurement tools for biology, including DNA sequencing technologies that have enabled rapid analysis of the human genome and microfluidic automation methods that allow scientists to isolate individual cells and analyze their genetic information efficiently. He previously served as head of science at the Chan Zuckerberg Initiative, or CZI, overseeing scientific strategy across CZI's research programs, technology teams, and the CZ BioHub network, and the Chan Zuckerberg Institute for Advanced Biological Imaging. If you have any questions during this talk, please type them into the chat or question and answer dialog box. At this time, it is my great pleasure to introduce Professor Quake.
Thank you so much, Dean. It's wonderful to reconnect and to be here. I'm very excited to share some of the things I've been working on in my science over the past, actually, couple of decades now. And particularly this question of where multicellularity comes from. So I'm going to start with sort of some very basic background in biology. And if you're very familiar with this, just remember what Mark Twain said: there's nothing ever so pleasant as thinking about something you already know, because we'll soon get into much more depth. But for those who are coming from a general audience, and it's been a while since you had some biology, let me just remind you that cells are really the fundamental quanta of biology. They're the smallest indivisible units of life. This is an image of a painting by the biologist and artist David Goodsell, who's done a whole series of these things. It's called The Last Universal Common Ancestor, and what he's done is try to describe what a dividing cell looks like, and he has, in as realistic a way possible, in the sense that he's taken the atomic resolution structures of the various components of a cell and tried to put them all in place. And what you see here are the lipids and sugars that make up the cell membrane. You see the innards of the cell, the various protein machines there that do the work of the cell, as well as that long piece of stringy looking things, which is the DNA or the genome. And these are and you get a sense for how densely packed the interior of a cell is. And so over the next hour, we're going to be discussing many aspects about how cells work by thinking about all these components and what they do. Now, the genome itself is made out of a molecule called DNA, and it is the molecule of genetic inheritance. This is what propagates information from parent to child, from one generation to the next, of inherited traits. And it very famously has the structure of a double helix, which is like two spiral staircases spinning around. And the steps of the staircase are these bases, of which there's 4 different chemical kinds. And they're often referred to by the first letter of their chemical name, ATGC. And they pair together. A is always opposite T, G is always opposite C. And together they encode the information which the cell uses to make proteins. And this structure was famously discovered in the 1950s by Watson, Crick, and Franklin. And if you're interested in the history of all this, there's very sort of classic books about the story of this. one written by James Watson, one written by Francis Crick, who were both principals. Rosalind Franklin tragically died young, and before she could tell her own story. But there's a wonderful biography by Brenda Maddox, and this is not sort of dry, boring scientists working away. There was a lot of emotion, intrigue, and it's worthy of any kind of thriller that you would read about. And it's interesting to get all their perspectives on it. It was a very complicated set of interactions. And when they discovered the structure, Watson went around saying they had found the secret of life. Because the structure of the molecule suggested many aspects about how genetic information is transmitted. And later, as they tried to work out the information flow and how it went from information in the DNA to synthesizing proteins by the cell, the field eventually arrived at this model, which Crick termed the central dogma, that the information in DNA doesn't go directly to making proteins, but it's copied from the DNA into an intermediate molecule called RNA, in particular messenger RNA or mRNA. And that RNA then is translated into proteins by the ribosome in the cell. And so for protein synthesis, it flows in the direction that the black arrows show. And when a cell divides, the DNA gets copied, and that's replication, which is the circular arrow on the DNA. So the information can be copied in new DNA molecules as cells divide and as you move from one generation to the next. And in some cases, you see the red star, the information can go from RNA back to DNA. That's done by certain viruses such as HIV through a process called reverse transcription. And so it took quite a while to work this out, but this is kind of the fundamental understanding of information flow at the molecular level for protein synthesis. And if you ask, why did I start with a picture of a single-celled bacteria, and why is there so much emphasis on these obscure organisms? It's that these processes are shared across all of life. They're really universal. Everything I've said so far is true of every organism. And Jacques Monod, who we'll come to again later, one of the great early molecular biologists, sort of had this very pithy phrase that anything true of E. coli must also be true of elephants. And so it reflects this search for universal aspects of biology that all organisms share. Again, another book reference for you if you're interested. This book by Horace Freeland Judson called The Eighth Day of Creation is sort of a beautiful history of molecular biology, written by, in this case, a dispassionate historian, which covers not only the discovery of the structure of DNA, but also the discovery of how information flows to the cell and the discovery of how those structures were determined. And it's a wonderful, wonderful sort of classic book. Okay, let's talk about DNA by the numbers. I hope I've convinced you it's rather important to study it. Every cell in your body has 2 meters of DNA in it. So even though it's a molecule, it is a very long molecule. You have your 23 chromosomes. If you laid them end to end in each cell, you'd have 2 meters of DNA. It's incredible. Each of you in your body has about 37 trillion cells, and every cell has 2 meters of DNA. If you do the math on that, every one of us has enough DNA in our body, if it was laid end to end, to go from the Earth to the sun and back more than 250 times. That's a lot of length wrapped up in our body. How do we do that? And how does it all fit? Well, the secret is, even though it's long in one dimension, it's very small in the other 2 dimensions. It's a molecule after all. And so the other 2 dimensions are atomic in their size and are only 2 nanometers in diameter. And that's how we managed to pack it all in there into very small volumes. This is a movie we made in my lab some years ago of DNA being manipulated using the force of laser beams to control beads that have been glued to the end. And you can see the molecule is long and skinny. This is imaged with optical microscopy, and we're tying it into a knot by making a loop and passing the beads back and forth through the loop. And this was part of a study to try to understand the effects of topology and statistical mechanics, but gives you a sense that the molecule is actually something you can see with your eyes. You can look under a microscope and see it using the tools of fluorescence microscopy. So, as a community, we've become very good at sequencing DNA, and the last couple of decades have been a revolution in the technologies behind sequencing. And you can see that here, sort of in this chart from Nature magazine showing the sort of massive decrease in cost of sequencing, which is shown in red as the cost per million base pairs of sequence is going down by orders of magnitude, as the amount of information being generated by sequencing is increasing dramatically over time, as the first human genomes were done in the early 2000s, and then the next individual genomes of various people were sequenced, including my own. So it's been an explosion of information. And now, let me just give you the opposite perspective. I've been trying to convince you how important the genome is, and it is important, but let me also say you don't want to overinterpret it, because people speaking loosely would sometimes call the genome the blueprint of the organism, but that's really the wrong metaphor. And let me tell you why. Pretty much every cell in your body has the same genome. I mentioned that earlier. Yet the cells choose very different identities. A neuron in your brain is very different than a cell in your lung, is very different than one of your muscle cells, and they all have the same genome. So how does that happen? The way to think about this, the metaphor is, maybe a better metaphor is that the genome is a parts list, and different cell types will pick and choose among the parts to create very different identities and functions. And so knowing the genome doesn't tell you everything, and it's not a clear blueprint, but it's a series of parts. And it's a very interesting open question in the field, and one of the mysteries of the cell, why I chose the title of this lecture: how do many different cell types arise from one genome? And how should you think about that? Is the genome a biological computer code which controls development in a deterministic fashion? There's many open problems interpreting the information content of the genome. In particular, it is still, after a couple of decades of having genome sequences in hand, it is still impossible to predict the various cell types of an organism from the genome alone. It's not clear that it's possible, in fact. Many of us believe it probably should be, but it's not been done yet. And so that's one of the limitations. And part of the research I've been involved in over the past couple of decades is to try to experimentally determine how the genome is used in these different cell types. And I'll tell you more about that. So here's a thought experiment. What if you could enumerate every molecule in a cell? Would this be a useful estimate of cell state and cell identity? Let's say we throw away all positional information and all the structures and how things are organized within the cell, and just make a list of the molecules that are there. I think most people agree that that would be, although not a perfect replica of the cell, provide an enormous amount of useful information about cell state and cell identity. Now, that's a very difficult measurement to do because there's many different kinds of molecules, but it turns out to be experimentally tractable, and I'll describe this in a few minutes, to actually do this for RNA molecules. You can count every RNA molecule in a cell. And in particular, we're going to focus on the mRNA molecules I mentioned earlier, which are the intermediates of genes being expressed and about to be turned into proteins. And sort of the question we've tried to ask over the past decade is, how useful is it to do this? Because we've gone and made these measurements, and that'll be the topic of the rest of this talk, is to try to explain how we did it and hopefully convince you that it's been biologically useful. Now, the way we've done this is through what's called single-cell transcriptomics, which amounts to amplifying the RNA from a single cell and then sequencing it. And this field has become, incredibly popular over the past few years. There's been explosive growth in the technologies, but it really goes back 3 decades. And it was back in 1990, just after the invention of the first biochemical amplifier, which is called polymerase chain reaction, or PCR, that Eberwein and Iskov independently worked out ways to amplify mRNA from single cells. And they did it by converting it into DNA. That's called cDNA. Remember, there's that backwards arrow in the central dogma that you can take advantage of. And then having amplified it, they lacked the experimental tools to probe the complexity of it. And so they were using something called a southern blot to look at one or two genes, but they really could not analyze the true complexity of what was in the cell. And it took another decade and a bit for people to develop technologies to do that. And in 2003, 4 different groups used what were called hybridization microarrays to measure thousands of genes from single cells. And that was a real breakthrough in understanding the true complexity of the molecular enumeration of these mRNA molecules in a cell. And then after that, people turned to sequencers to read out the molecules. And that's how we do it today. We use these next generation sequencers that were used to sequence the genomes, also to sequence these mRNA molecules from single cells and to do many molecules from many cells. So this sort of describes the evolution of that. The piece that is missing here is the ability to do this at scale on lots of single cells. These early works were all sort of bespoke experiments with a handful of cells, and what was missing was a way to do this at scale, and that's how I became involved in my research with this field. I had been working on trying to build tools to automate biology, and we had developed what are called microfluidics, which is essentially miniaturized plumbing to do biological automation. And one version of that was sort of very literal interpretation of how integrated circuits are made. With integrated circuits, automate math by using batch fabricated wires and transistors and such forth. And we did used similar techniques to build chips that had tens of thousands of micromechanical valves and pipes and pumps on them. And my group had been exploring the applications of this in a variety of different areas of biology, including single-cell genomics. And a company I founded called Fluidigm built the first commercial device to do automated single-cell genomics. It was called the C1, and it had the amazing throughput of 100 cells per chip, and it really launched the field and allowed any biology lab to use these techniques at scale to ask questions. And it was sort of a real opening of the field. And there's a lot of interesting sort of engineering behind how you manufacture these chips at scale. And the company at the end, made billions of valves. A truly phenomenal engineering achievement. But as people adopted these techniques and used them, their thirst for data just grew greater. People wanted a higher throughput. That led to a second generation of microfluidic approaches to do single-cell transcriptomics, again, based on work done in my lab quite some time ago, in which we created microfluidic emulsions on a chip, so sort of droplets of water and oil, kind of like a salad dressing. With that fundamental tool, a group of faculty in the Boston area developed a version of single-cell transcriptomics in which cells and beads were captured in those droplets. And those were commercialized as well and allowed one to do thousands of cells per chip. And so the basic sort of workflow for these types of experiments became take a piece of tissue that you cut out of an organism, you chemically digest it so all the cells are in suspension, you put it through one of these microfluidic devices to isolate the cells, break them open, amplify the mRNA, label it, and then you sequence it all. And every cell is barcoded with a different sort of DNA barcode, so you can mix it all together, sequence it, and then deconvolve it at the end, and then you compute on all this data. And roughly speaking, a human or a mouse has about 20,000 protein coding genes. So every cell is embedded in a 20,000 dimensional space, where each dimension represents one gene from the genome, and the number in that vector represents the number of mRNA molecules from that gene that were expressed in that cell. So it's a very sort of rich data set. And there's many interesting sort of computational challenges in analyzing it. So when I first started working with the Chan Zuckerberg Philanthropy, my goal was to try to put together a very large collaborative science project to use these techniques to characterize all the cell types of an organism. And that's why we founded the Chan Zuckerberg BioHub in the Bay Area, which I led for the first decade of its existence. And we assembled a large group of people to do this work. And it was terrific team science. These were papers that had 150 authors on them, and many, many people contributed. we made a cell atlas of the mouse that we called Tabula Muris. That led to us helping an international collaboration to do the fly, which is an important model organism. We then also used our mouse consortium to do the lemur, and then ultimately human, Tabula sapiens. And this work, I would say, has led to good rough drafts of whole organism cell atlases. We wouldn't claim to have every cell type, but we have many, from 18 to 24 tissues, depending on the organism, and a very sort of high quality characterization of the data. Part of the way we structured these projects was to recruit experts of each tissue we were analyzing, and they not only helped do the wet work of analyzing the tissue, but also the informatics. And we had expert human curation of every cell that came out, and that established a good ground truth for cell type. Another aspect of this, which I'll get into a little bit more, is we worked very hard with mouse, lemur, and human to get multiple tissues from the same donor for reasons of scientific quality, data quality, and control. And I'll talk about that more in a moment. So just to give you, I'm not going to get very deep into the biology here, but these papers have been published, and I'll point you to the references. We started with mouse. We were interested in aging effects. Tony Whiskaray, who's a colleague at Stanford and had been a collaborator on this, had been growing mice in his lab up to 30 months of age, which is very old for a mouse. It's equivalent to 100 years for a human. And he's very interested in questions of aging. And we decided to look not just at the definition of cell type, but to see how the gene expression was changing with age in mice. And Spiros Darmanis and Angela Pisco at the BioHub were key leaders of this. And we published these papers on natural aging of mice. Again, I won't dwell on it, but you're welcome to read it and see some of our scientific conclusions. We also wanted to explore another effect that Tony and his colleagues have been studying, which was ways to reverse aging by having young mice and old mice sharing the same blood supply. So they were surgically attached to each other to share blood, and it is through a process called parabiosis. And what had been apparent in people doing this work is that when you provide young blood to an old mouse, aging is reversed, and that old mouse starts to display characteristics of a younger mouse. And similarly, the young mouse starts to have unnaturally accelerated rapid aging. And so we did a molecular dissection of that. That was very interesting. We could compare that to natural aging, and we learned a ton about that. I'm going to dwell a bit longer on human now, our Tabula sapiens project. Again, we had a large consortium. I'll just acknowledge Bob Jones at the lower left, who sort of quarterbacked the whole thing, but it involved many people from Stanford, UCSF, Berkeley, and was a real team effort. With the human work, we not only did the single-cell transcriptomics, but we also got pathology, and in some of the tissues, the microbiome. And the first version of this was about 500,000 cells. We've now increased it to 1 million cells, and you can see here sort of how that's distributed. So every row here is a different tissue. Every column is a different human donor, and each symbol tells you roughly how many cells of that tissue we got from each donor. And you can see that we worked very hard to get many tissues from a single donor. And we did that in part because we wanted to control for the genome. We wanted to have the same genome in every tissue so that we can compare cell types across them, as well as controlling for age, environmental exposure, epigenetic effects, and so forth. So part of the special nature of this work was the ability to get many tissues from the same genome. And it led to the expert annotation, led to more than 700 tissue cell types. And I'm going to tell you some more about what we learned along the way. But first, let me tell you a bit about where the donors came from and where the tissue sources were. We worked with a terrific collaboration with an organization called Donor Network West. They are a nonprofit organ procurement organization that serves roughly 13 million people in Northern California and Nevada to help those who need organ transplants to save their lives receive them. So people who have agreed to be organ donors and then have had some sort of tragic accident This is the organization that connects those people with the people who need to receive the donated organ. And in many cases, the organ donors have also consented for there to be research with their tissues. And it's with those very generous donors that we were able to do this work. And we're very grateful for people who have volunteered for that. When it happens all at once, and it requires basically an all-night fire drill. The surgeons are working to take the organs out one by one. First, the ones that are going to be transplanted are sent to the people who need that, and the remaining tissues, if it's been consented, are given to us. And our folks are waiting to courier them to the expert labs, where they're then processed and turned into molecular sequence data. And it requires an enormous amount of dedication by the team of students, postdocs, and staff members who are doing this work. Much of it was done through COVID. You can see they're wearing masks, and it was just really an amazing sort of dedicated team of folks, and their effort is what made this all possible. This is a list of many of the names, the people involved. As I mentioned, it's a tremendous team science project, and it required coordination far beyond what gets done in a single research lab. So what did we do with the data we learned from these experiments? For that, let's talk about the so-called second secret of life, which is something that was discovered by Monod and Jacob. I'd mentioned Monod earlier. Here they are in their lab in Paris. And one of the things they discovered is this question of what regulates which genes are used in which cells. And they discovered that some genes actually control other genes, which is known as regulation. And that was an amazing discovery about information processing in the cell. And over time, we've learned how this works. And basically, the way it works is that proteins will bind to the genome. They'll bind to the DNA at the beginning of genes, and they will either turn on or turn off those genes by recruiting other proteins there. In this case, transcription factors are proteins that will bind to the gene and recruit the machines that copy the gene into RNA. And that machine is called RNA polymerase, and it will come bind to the genome in places where there's transcription factors signaling it to come turn on and copy that information. And so understanding this goes a long way towards answering this question of how the parts are used in different cell types of the body. And while this was known in principle as the mechanism, the details of which transcription factors control which cell types were not known until we did this work. And so we looked at all human transcription factors. The community has identified about 1,622 genes that are putative human transcription factors. Some of these have been well studied and have been proved to be transcription factors. Others are theoretical hypothesized transcription factors based on their sequence similarity to other transcription factors and have never been studied. So we went through and looked at every single one of these and were able to discover which cell types they were expressed in. And now we know for the first time which transcription factors are used in which cell types across the entire complete list. And this is a representation of that, showing a subset of them. The way to interpret this figure is each column is a different transcription factor, each row is a different cell type. And you can see for the majority of this diagram, most transcription factors are expressed in only one or a handful of cell types. So they're very specific, and in fact, they tend to define cell type. And this work provides a map of which transcription factors define which cell types. And when you drill in some very well-studied cell types, you can see that we recover the transcription factors that were previously known for those cell types, as well as many new ones for cell types that have never been described before. If you look at the rightmost part of this diagram, where it says 162, those are the 162 most ubiquitously expressed transcription factors. And you can see that those are expressed in every cell type. And these are transcription factors that are used for basic processes that every cell uses that are independent of the identity, but cells need to do. And here are some examples of that. We found many in that group, we found many well-studied transcription factors that are part of universal cell functions like stress response, proliferation, cell division, metabolism, All of that every cell needs to do, and therefore those transcription factors are used in every cell. However, in this list of ubiquitous transcription factors, we found a number of previously uncharacterized transcription factors, and particularly these zinc fingers that tend to have names of Z and F followed by a number. And so it's amazing, despite nearly a century of molecular biology, there are transcription factors that every cell uses that nobody knows the function of yet. And this work has helped identify some of those, and I think will point towards some very interesting discoveries of basic cell biology that has yet to be discovered. Okay, let's talk about another aspect of the data that we analyze, which relates to aging. I'm going to give you a little bit of background here. There is something called the Hayflick limit that was discovered or articulated by a fellow named Leonard Hayflick, who discovered that cells are not immortal. In his day, there was quite a bit of controversy of whether cells could divide forever or not. And the sort of prevailing wisdom was that cells were immortal. They could divide in artificial culture forever. It turned out that was not true. And the experiments that seemed to indicate that all had flaws. There was contamination and things like that skewed the interpretation. And Hayflick did the first really careful experiments that didn't suffer from those flaws. And he discovered that normal cells can only divide 40 to 60 times before they stop dividing. And then they're done. And they enter this state that he called senescence. And senescence is something that's now been very well studied. It's associated with aging. Many of the molecular details of it have been worked out. It's an area of intense interest. And we're going to talk about it in a moment. But the Hayflick limit has an interesting corollary. If you believe there's only roughly 50 cell divisions, and you know how long a cell division takes, you can estimate the maximum human lifespan. You know how many cells are in the body. That turns out to be 120 years is the theoretical lifespan. And that's sort of more or less what people observe. And Hayflick passed away 2 years ago. In his obituary in the New York Times, they had some fun with the headline, which read, "Leonard Hayflick, who discovered why no one lives forever, passes away himself." So we began to study the presence of senescent cells in our Tabula sapiens atlas. We realized we had probably the largest collection of naturally occurring human senescent cells that anybody had studied, and we had it across many different tissues. Madhav Mantri, who's a postdoc in my lab, began looking at senescence. Roughly 3% of the cells, 3-4% were senescence, which is consistent with people's estimates of the presence. For those who care about the molecular details, we define senescent cells as those that were CDKN2A positive, MKI67 negative, and began to look at their gene expression programs. And Madhav asked the question, is there a single universal gene expression program for senescence? If you compare senescent cells to their non-senescent counterparts of the same type and look which genes are expressed at different levels, are all senescent cells the same? And it turns out they're not. And in fact, there's no single gene expression program that is shared by all senescent cells except for the one we use to define them here. And that was a very interesting discovery. Then we began to ask, are there certain common denominators of classes of genes that are shared between senescent cells? And yes, we found a set of broad classes of senescent cells that are shared across tissues and use some aspects of shared gene expression programs, although not the same ones. So there's about 16 different broad classes of gene expression pathways, and each type of senescent cell draws from that in different amounts and proportions. And we've been able to articulate that in a fairly precise way. So I hope from this I've managed to sort of convince you that these large-scale atlases of single-cell transcriptomes are really phenotypic companions to the genome that help us interpret how the genome is used in different cell types by giving us this molecular definition of cell type. There are many, many interesting biological questions one can ask with these atlases. One that I don't have time to get into today is, does identity as defined by the transcriptome relate to identity as described by physiology, which is how people used to define cell types before we had all this genetic information? The answer is yes, in large part, and we did a lot of work to prove that. And I've given you a sort of flavor of some of the basic biology questions we've asked and answered with it. But now I'd like to tell you a little bit about how we're trying to synthesize all this data together. Because our group, the BioHub, was certainly not the only one working in this field. We specialized in doing whole organism cell atlases, but other groups were working on particular tissues. And in fact, at CZI, we funded much of that work around the world and then collected all the data in a single informatic resource called Cell by Gene, which is freely available for anyone to use. You don't need to be an expert. There's a web-based interface to browse around. When I made this slide, there were 33 million cells. There's now more than 100 million cells in the data set, and we expect that to grow over 1 billion cells over the next year or two as the community continues this work. And so there's a very natural question about how do we synthesize all this data into one sort of object we can query that represents the manifold of all molecular definitions of cell types. And we decided to try to do that using the tools of artificial intelligence. And as you all may appreciate, we're living right through an amazing revolution, which is both amazing and scary at the same time. If you've played around with any of these image generating tools like DALL-E, you can give it a phrase of text and it'll generate a picture for you. Here's some phrases I use to generate pictures. One of them, a very realistic set of people walking on the beach. Another one, I asked it for a painting in a certain style of all the proteins in a bacteria with DNA nearby, and you see that in the middle. And then the last one, I asked it to make a picture of a cell atlas in the Impressionist style. And it came up with this very sort of beautiful abstraction of what it thought a cell atlas might look like. Those sort of results are achieved with what are called large language models. And we tried to apply that same sort of approach to the analysis of the molecular data in the cell atlases. And so we trained a large language model on the data. And we did it in the same way that the models are trained on text, in that you cover up some of the data and ask the model to predict the missing data, and then you sort of continue to refine and optimize coefficients in the model to do that until it does it as well as it can. And this is work done in collaboration with Yuri Leskovich, a computer science professor at Stanford, and I'll talk about the students on the next slide. But this was a 600 million parameter model trained on 36 million cells. Over the course of about a month. One aspect of the design that was, I think, very powerful was to not use the genome to represent the cells. And in fact, we used what's called a protein language model, which meant that our large language model was not dependent on any genome, and it was designed to work with all species, not just human. That, I think, is a very powerful aspect of trying to synthesize data across evolutionary history. We trained the model initially on 7 or 8 species: human, mouse, lemur, fish, pig, other primates, and frog. When I say we, I mean 2 very talented students, Yusuf Rouhani and Yane Rosen, who were actually doing the heavy lifting on this. And you can see across those 8 species, it's primarily human. Next most common is mouse, and the other species are there at a lower representation. But as you'll see, we proved the principle of looking at multiple species. And this was roughly 50 tissues, 300 data sets, 1,000 cell types. So first question you ask of a model like this is, what does it do with new data it's never seen before, that's never been trained on? And so we had withheld the second half of the Tabula sapiens data set from the training. When we then tried to embed it in the model, after the model was trained, we saw that all those new cells went to the right place. They went to similar cell types and organs, and it agreed with all the expert annotation we've done. And so the model was able to use what's called out of distribution data, what wasn't trained on, and it performed well with that. So this is maybe the easiest out of distribution data because it had been trained on human, but this was human data I hadn't seen before. We then gave it a harder problem. We gave it a species it had never seen before. In this case, a data set on green monkey. And when that data set was embedded in the model and we asked what cell types were closest to the green monkey cell type, the model would predict which cell type they thought it was. And it did a very good job in pretty good agreement with with what the authors of this work how they had annotated the cell types. And so this demonstrated the model could analyze data from species it had never seen before and do a reasonable job at that. We began to look for emergent behavior in the model. We didn't train the model any biology. All I got was a matrix of numbers, cells by genes, and just those numbers, didn't have the names of the genes, didn't know any of their biology. And so as we began to measure distances in the embedding space of related cell types, we saw that all the immune cells were close to each other, and subtypes of immune cells, like T cells, were particularly close to each other. Muscles were close together. Endothelial cells were close to each other. So the model had learned something about the underlying biology of these cell types without being taught it at all. And that sort of emergent behavior made us feel that something very interesting was happening. And we did a more general analysis of that by using the cell ontology map, using what we knew about the lineage of cell type development to look at parent-child, sibling, cousin relationships. And we found the more distant the relationship between cell types, in the most general sense, the more distant they were in the embedding space. And so the model had learned something very fundamental about lineage and development of an organism in relationship to cell types without us having explicitly taught it. And we're going to go on now. We've already built a second generation of models at CZI called Transcriptformer, which is exploring these evolutionary distances in much more detail and ambition, covering more than 1 billion years of evolutionary distance. That paper is up as a preprint if you'd like to look at that. And I think the work in this general space is only going to accelerate as these models become more refined and more useful. In the last part of the talk, I just want to say a little bit about some of the medical applications of the work. We've been talking about basic science all the way along. I'll give you a couple of quick examples of how this work is being used in the medical field. The first is a diagnostic example. We have been interested in using what's called cell-free RNA to build diagnostic tools. And cell-free RNA is RNA in the blood that turns out every tissue in your body, when cells die, and sometimes even as part of their natural physiology, they'll contribute RNA into the bloodstream. And so, in fact, your blood contains RNA molecules from every different tissue. And that's a powerful basis for so-called liquid biopsy diagnostics. And a student in my lab, Savan Verparian here, decided to try to use the tabula sapiens to deconvolve all the cell types in the cell-free RNA and to try to understand what contributed to the mixture. And so she did that. I won't do all in the details, but worked out the pie chart of how many different tissues are contributing. Many of them, the largest contributors are from the immune system, because that's what's mostly in the blood, but many of the solid tissues are contributing as well. And she worked out the proportions and ways to, given this mixture of RNA, to work out how much of each cell type in the body is contributing. And she was able to find that in cases of disease, she could identify cell type specific changes, both in fatty liver disease and in chronic kidney disease, find signals that seem to indicate at the cell type level what are the differences between healthy and sick people that could become potentially someday the foundation for new diagnostic tests. We've also worked on using these techniques to develop therapeutics, and this dates way back to the first development of these techniques, even before the highest throughput versions that were used to make these cell atlases. Mike Clark and I were using these techniques to look at tumor heterogeneity in cancer to find drug targets. That led to several new small molecule drugs that are in various stages of human trials to cure cancer. The story is not yet complete for them. Drug development is a lengthy and challenging process, but it's gone quite some ways from basic science into real clinical medicine, and hopefully these drugs will be saving lives at some point. As another example of therapeutics, we began to look at people with food allergies. This is an immunological example. And Derek Krute, who's a student in my lab, and Kari Nadow, who's a collaborator, and was running the allergy clinic at Stanford, we began to look at the blood of people with food allergies and look at the cells that cause the allergy and produce the antibody that gives anaphylaxis. And in analyzing that, Derek made a number of discoveries, not least of which was the identity of these antibodies and their sequences, which we could then express and study their properties, and as well as the fact that different individual patients had evolved exactly the same antibody, which was an amazing discovery. Here's an example of this sort of conversion evolution, very highly similar antibodies in two unrelated individuals. This must be doing something very important, have strong evolutionary pressure. And sure enough, these are the ones that bind to peanut and cause the allergic reaction. And knowing that, we then began to study their affinity and their biochemistry. I won't dwell on that. This has been published, but began to do larger scale studies in the context of a company we founded, reproduce this result of convergent evolution across much larger numbers of samples and individuals, now thousands of sequences, and hatch this idea to create a therapy that would cure people these food allergies. And we decided to try this idea of re-engineering the antibodies to get rid of the part that causes anaphylaxis and put a protective sequence in there, and then administer these antibodies to people to compete with the bad antibodies and potentially neutralize any situation when the person consumed peanuts and provide protection. And Derek and colleagues at the company were able to do some work with mouse models to show this works in mice as a potential new therapeutic approach. It's now in human trials, and later this year we'll see the results from that and hopefully have some good news. So with this tour, hope I've convinced you of the kind of power and value of doing this very basic science study of molecular characterization of cell types, and not just humans, but many other species. And we're beginning to make not just fundamental biology discoveries with that, but also develop things that will be powerful tools for human health. Thank you, and I'm happy to take any questions right now.
Thank you, Steve. That was beautiful. Really, really interesting science that you're doing, and very, very important. So that was wonderful. There's a question. Yeah, so if other people have questions, go ahead and post them. There's a question from Gordon Push, who asks, I mostly work with prokaryotes rather than eukaryotes. The myxobacteria appear to behave much like eukaryotic slime molds. Do you look at myxobacteria as models of cellular differentiation?
Oh, that's a great question. There are very interesting models of, multicellularity, not so much with bacteria, but with other single cell organisms. There's folks who have managed to get yeast to combine in complicated ways that look multicellular and study them. Choanoflagellates seem to move between single cell, multicellular versions and sort of cooperative things. And so definitely there are models there. And prokaryotes, which come together to create biofilms, have some aspects of that as well. It's a little more distant, but I think provides intellectually an interesting model to get at questions of multicellularity.
Mm-hmm. Okay. There's a question from Sudin Bhattacharya, who asks, can the networks of genes you mentioned predict the diversity of cell types?
Well, so, there's a question of what's predicting what. And I'm really glad you raised that question. Because we've been unable to use the genome to predict the diversity of cell types, we set out to measure the diversity of cell types. So I've described the measurement of it. And, your question is getting a very good point. Can we use these measurements now to build predictive models of cell type diversity of genomes that haven't been studied yet? And the answer is, I believe we can, and that's why we've begun building these large language models, because ultimately we want to turn them towards predicting cell types from novel genomes that haven't been studied yet.
Okay, if there are other questions, go ahead and post them. Maybe I can ask a question. I have a question about structure and function. So if you have a given segment of the genome, is there one-to-one correspondence between that structure and a particular function of what it does, or does it have multiple functions? I'm trying to understand how sort of oversubscribed this information is, that it's doing multiple things at the same time.
Yeah, sort of the biology textbooks will say one gene, one function, yeah. But it's actually much more complicated than that. And, we understand now that many proteins will play multiple roles biologically. And so it's an oversimplification to say one gene, one function. And this is, something that is interesting, not just as a basic science question, but also practically, because when you start to drug things, sometimes you get unintended effects because you realize these proteins had other functions. So yes, that's a complicated, multi-layered question.
Okay. There are a couple of questions here. Jianliang Chen asks, thanks for the talk. How does the three-dimensional genomic structure influence the cell type evolution?
Okay, another very good question. So the genome has a lot of structure. It's physically organized in a lot of structure spatially. And there's parts of it that are brought close together in various parts of the cell. They tend to be, like even in bacteria, the genome is not randomly distributed, but there's very sort of stereotyped positions in the cell. And people have begun to map that out in some detail. I think some of the reasons are understood, some are still very mysterious. But for sure, there is a connection between geometry and gene expression that we just, are just getting our heads around, and I would say don't fully understand. I at one point began giving lectures. The cell is a bag of RNA, and part of the theme of the lecture was how much can we learn about cells by not just ignoring the structure of how things are organized in the cell. By that I meant not just the genome, but also the organelles and things like that. And it was meant to be a play on the early biochemists. They viewed the cell as a bag of enzymes, and they tried to reproduce the chemical behavior of these enzymes in test tubes by ignoring the structure within the cell. And while those structures are important, you can learn a lot from sort of averaging over that. Let's call it that.
Okay, a few more questions have come in. Margaret Chung asks, is there any interest in working with the Joint Genome Institutes or other DOE national laboratories where data types are abundant for non-human related research to explore genotype to phenotype research?
Oh, absolutely. Sort of the Joint Genome Institute in particular has been an absolute pioneer in microbial genome sequencing, has established one of the largest databases for that. I worked with them very early on around some interesting questions of the human microbiome. They're a terrific organization, and it's been just sort of a phenomenal group. And also, I will say that the JGI was involved in early sequencing of the human genome. So they have done work in human genetics early on and moved on to plant genomes and things like that. So they remain very important contributors to the field.
Okay, very good. Let's take a couple more questions. So Tom Halloran writes, nice talk. It looked like there were more senescent cells in females than males. Is that the case? And if so, why?
Yeah, I wouldn't say that we have the statistics to really draw that conclusion. And we have a relatively small number of donors. Even trying to analyze the number of senescent cells as a function of age has very large error bars for us right now. And so that's going to require a higher end to really be definitive on the statistics.
Okay, Dennis Disher writes a short question. RNA splice forms in phenotypes, question mark. Major or minor role, question mark.
Huge role. Okay, so I sort of glossed this over a little bit, but this idea that when a gene is copied into RNA, that there's only sort of one transcript per gene, that turns out to be an oversimplification. And it turns out in eukaryotes, the gene is not contiguous in the genome. There's different segments that then have to be spliced together. And the splicing can happen with a certain amount of combinatorics. Sometimes regions are dropped out, others are added. That's a whole nother sort of level of complexity. And typically any given gene has between 2 and 5 splice variants. So that increases the effective sort of diversity of protein products that can be made. I didn't talk about it in this talk, but we have analyzed that in the first Tabula Sapiens paper and discovered many new splice variants that are specific to certain cell types. If you go back and look at that paper from 2022 in Science, you'll see a long discussion of that. And it's a very interesting question.
Okay, maybe I can ask one more question. So when I think of evolution, I sort of think of success strategies for different organisms, right, competing against each other. But do you see in your data evidence of evolution within one organism, different cells competing against each other in some way to have better function?
Yeah, I mean, those sort of ecological questions of evolution, there has been a large literature about that, about whether those principles apply within the human body. And did they come into aspects of develop multicellularity? And in particular, do they have a role in cancer? where the cancer cells are trying to cheat evolution by dividing without restraint. So I will say that those questions have been posed. There's literature about them. We have not done a deep dive in our data yet about them, but that's why the data is there for those scholars to try to make use of it.
Okay. Well, thanks so much, Steve. This was wonderful. I think at this point we will end the public part of this talk. So if you want to stick around and just ask Steve some questions privately, feel free to do so. Otherwise, thanks everybody for coming. Have a great weekend. I'll see you guys next time.